See, nobody asked me for online presentation. I am
Question 3 on potential of gene editing and genetic engineering. We are greatly honored to have with us a panel of professionals well versed in this field as well as the participants. We all know that in recent years, gene editing technology has revolutionized the field of science, emerging as one of the humanity's most powerful technology in the pursuit of great understanding of diet modification and more specifically human health. So gene editing is a technology which allows us to target almost any gene for modification in a very specific and very reliable way. So today we have with us a panel of professionals well versed in this field and who will discuss and throw some light on the potentiality of this technology, the latest developments, challenges in using this gene editing technology. So for this panel discussion, we have the panelists, Dr. B.G. Malati, former principal scientist, IARI, and we have with us Dr. S.R. Bhatt, former emeritus scientist, IARI, Dr. B. Fakhruddin, Professor and Head, Division of Biotechnology and Crop Improvement, College of Horticulture, Bengaluru. We have with us Dr. K.S. Mohan, Advisor, Biotech Product Development and Stewardship. Sir has joined the session online. And we also have with us Dr. Ravindran, Director of Research from TNAA Coimbatore. Sir has also joined online for this session. And also we have the panelist Dr. Y. Suresh Reddy, Senior Scientist from IBR Varanasi. And for this session, the moderator for this session is Dr. Nataraj Karpa, Professor of Plant Physiology and Biochemistry, GKVK Bengaluru. And the rapporteur of this session is Dr. B. Fakhruddin, Professor and Head, Department of Biotechnology and Crop Improvement, College of Horticulture, Bengaluru. I request Dr. Nataraj Karpa sir to moderate the session and to take over the session. And we have a lecture by Dr. E.G. Malati madam initially. After that we have the panel discussion.
Apologies for kind of delay. We were planning this session in 4
Mic isn't working.
not all.
Time being also, we need to remove the chairs, right?
Yeah,
so that we can see the screen.
Since we have limited time to debate and discuss, I don't take much time to introduce the topic. Everybody knows about this. I'm very fortunate and lucky that organizer asked me to coordinate this particular session on gene editing and genetic engineering. And this particular topic, all of you are aware, that is hot topic now, and we must recall the contributions of the Carpentier and Downa, Jennifer. So they discovered and actually developed this into a kind of method for genome editing. Therefore, we are all using this particular technology now for our, say, targeted manipulation of characters, traits, especially in plants or the genotypes or the system where there are limited options for genetic manipulation and genetic diversity is very low. So since we don't have much time, I don't want to go through this video. This is actually for the beginners who are not aware of this particular technology. So we have some kind of the animated videos to give an idea that how best we can manipulate the genome when there are less option for us to use traditional breeding approach. So I think we will have eminent speakers who will, or the panelists who will tell us more about that. So the technology what is available with us now can be used for manipulation of characters, diverse type of characters, especially in horticulture crops. I am looking only towards horticulture today, starting from ornamental to all kind of fruits and vegetable manipulation. In fact, it started, if you go through the literature, which I borrowed from some literature in the morning, from 2016 onwards, people started playing the different types of horticulture plants, see whether we can create variability and also have a targeted manipulation of traits required for either for fruits and or nutrition angle or for ornamental purposes in different crops. This is just an idea, but today's discussion will be more focused towards tomato because every speakers today and yesterday also debating on only tomato and tomato. So you know that in conventional breeding we have long time to generate the genotype or the variety of our choice, and genetic modification is possible through transgenic and cisgenic based approach. Again, we have certain regulatory issues associated with this. Now we have this genome editing technology, and we are thankful to Government of India for allowing us to have some kind of edited versions of the plant types for our so-called crop improvement as well as commercial usage. So we have now two types of things exempted from so-called GMO regulations. Exemption is SDN1 as well as SDN2. However, the SDN3 is still under regulations. So in simple, government helped us to move ahead with genome editing technology to create variability in the germplasm as well as for targeted manipulation of traits. And everybody, those who are working in this domain, are aware of this. So in tomato, in particular, why are we debating with respect to genome editing is because of narrow genetic base in tomato, and that is a major challenge for most of the breeders, especially for trait improvement as well as viral or disease resistance. So therefore, this genome editing, especially whenever I see the literature, I see this CRISPR-based technology being extensively attempted in tomato system. And I went through this review of Tiwari et al. They have tried to summarize the activities that are happening with respect to genome editing in tomato. So plant phenotype and fruit yield related, several genes have been identified. Fortunately, we have genome information available, and we have a handle to play with the architecture of the plant phenotype as well as the fruit and nutrition as well as quality related traits can be manipulated. This is just a list of the genes which they tried or attempted. Some of them may not be useful, but then attempts have been made. Not only that, for improving the stress resistance, basically biotic stress resistance, people have attempted and experiments are going on. And we do have a problem with respect to several types of abiotic stress, mainly salinity and drought and high temperature related issues. So now there are genes or candidate genes which can be tried to activate the trait. This is just a summary of the overall thing which is available in the literature and summarized extensively by Tiwari in this particular paper. So before I conclude and hand over to my panel member, I just want to tell you that the GABA enriched tomato is the first CRISPR edited tomato which is available in the market or launched for the market. That's what the literature says in Japan. So you do have now this kind of possibility of improvement of nutrition with respect to tomato, and I hope we will be focusing more and more on this kind of very important crop. All of you are aware of that. TOP are the important crop for us: tomato, onion and potato. So the tomato is the top in that. So people will be interested to work further on this, and I'm sure this particular debate what we are going to have will have a lot of options for us to look for the precision breeding. In case those who are interested in disease resistance, there is a possibility, and targeted improvement to traits as well as evolving genotypes, plant types with more climate resilience and also germplasm conservation angle. So with this background, I request the other panelists to continue with the discussion. Now we have, as per the list, although we may not go through the list, I request Dr. V.G. Malati to take over and give an outline and help us to discuss further. Over to you, ma'am.
First, I think I should thank all of you for assembling here, because I was almost expecting none here, and I thought it would be a free go for me. Thanks a lot for coming, and that shows the support that you always give for me. And there was a lot of confusion. Dr. Tushar was expected to give the complete technology and the basics of this technique and all. But I don't know what happened, and suddenly I received a communication for preparation. I have tried my level best, and let us see how it goes. As I had correctly put it in my second slide, it's a first disclaimer. I have no hands-on experience. And so before you all people pick up the microphone, to talk, to ask me questions, just have some consideration for me. I'm just like any one of you, trying to learn by just reading and reading, and especially Surendra Tikuji and Sharan Angadiji made me work last 1 week. So I had studied and then I had tried to prepare something, and I hope it will be interesting. Next is please. As our moderator, I'll be just introducing the technique, and then I'll be quickly going through the resistance that has been generated against oomycetes and fungi, and then bacteria and viruses. And finally, a very good paper, mainly for Dr. Tikuji, that how we can use this genome editing technology for domestication of the wild lines. And that is something, a dream of a breeder, I would say, that you can bring in one sort of exercise, having all the gRNA targeting the unwanted lines. It has been worked out, and let us see how it goes like that. Next slide, please. Next slide.
Next slide.
What is the committee? So this is, there are 3 different types, and the one that is being very routinely used is the CRISPR-Cas. Next slide. Of course, the Nobel Prize winners. The very peculiar news about this is that she was awarded the Nobel Prize in her house, and that particular YouTube was very popular, that it was shown how in that backyard garden she was awarded the Nobel Prize. Next slide. So the basic concept, you can completely do
Sure.
the basic concept, as Dr. Natarajan had clearly mentioned, is that it is all from the prokaryotic genome, that the bacteriophage DNA is cleaved by the enzyme endonucleases, and the cleaved sequences are all stored in a CRISPR or in a repeat sequences like in the genome of the bacteria, which is associated with the endonuclease Cas9, but there are about more than 23 families of Cas endonucleases which have been characterized, and they always are associated with their scaffolding RNA sequences also. So this is the exact thing happening in the bacteria, that from these spaces or the CRISPR sequences the transcript comes, and the Cas9 is transcribed and translated, and the transactivating cRNA is also transcribed, and together they form the complex, and the complex go and attack the bacteriophage DNA when it is coming inside the bacteria. So this is a very, very short concept, but this is being exploited. Next slide, please. And on the left shown is what is happening in the bacteria. So on the right, what we are doing it is that we synthesize a single gRNA, and that gRNA will be having the target sequences that you are going to edit. And then we are going to have the commercial vectors giving you all the things like the different types of Cas proteins, the different type of Cas scaffolding sequences, etc. and all they give. So all the job that you have to do is decide on what is the gene you are going to target. And the gene that you are going to target, it should have that protospacer motifs, PAM, NGG, it is usually the NGG motif for Cas9, and that sequence should be having that PAM sequence or the NGG sequences. Accordingly, you have to synthesize your RNA, and you give the sequences to any of the company, and they will synthesize it and put it in the vector and give you. Next slide, please. So this is the technology. This is what I have explained again, that you make a single gRNA which will have the scaffolding transactivating cRNA, and the spacer you have to design on your own, and the Cas9 is also present in the vector, and that you use for your treating your plant cells. And then what happens is the Cas9 binds and the cleavage takes place, and then it gets repaired either by homologous target recombination, or you can have any insertion or deletion depending upon what is the type of experiments that you are planning to do. Next slide, please. So today I thought, like since there are many experts and then physiology experts are there, they would be talking about the traits of quality, yield, and plant type and all those things. So since my specialization is pathology, I thought I would talk only on how you can use the GE for generating disease resistance. So the two major approaches, as you see, is directly you can target the pathogen genome. And that is possible, and very, very rare cases it has been done, and that we will discuss it much later. Whereas the one is the target the gene whose product is helping the pathogenesis. That is what is a very important thing. You should know what is the factor which is causing susceptibility in plant. And this is totally a shift in our approach. Because so long we have been studying only resistance, and we are very clear about the gene concept, PAMP, MAMP and all, and what is the resistance mechanism we know. But what is the susceptibility mechanism, very few cases have been worked out. And if you look into it, most of the cases where a resistant plant has come is that particular susceptible factor would have been mutant. So these are all actually mutants and variants which are naturally occurring. It's like millions of such mutants and allele variations may be there in the plant. It is up to us to scoop for it, which is very difficult. But even in these experiments, initially what they did is that, okay, for example, we know that the elongation factor is the one which is contributing to the susceptibility of both potato viruses and the cucumber mosaic viruses. So there were a lot of projects in DBT where people were looking for the natural variants of susceptibility factor which would be having an altered sequences which would not support the pathogen. Then once you know that by such mutation the susceptibility factor is becoming or conferring resistance, you can use those factors. But instead of mining for those kind of mutants and variants, there were possibilities of creating mutation, but earlier days we had gamma rays, we had EMS, and then creating mutation and then sorting out the mutants also took a lot of time. So now we have this technology of genome editing. But you should know the factor that is contributing to the susceptibility. That is very, very important. Next slide, please. So the technique is not that easy as we all make it out to be. For example, first is you have to know what is your target gene. The second thing, you have to design your gRNA. Like, of course, there are softwares, and then you have to sit and work on the maps, and then you will definitely come out with the most conserved sequences of the susceptibility factor nearer to the PAM. That way you can have visualization of what is RNA that you are going to have. And then you get it synthesized. And you put the whole thing in the vectors, and there are thousands of vectors available commercially with different types of Cas9, and each Cas9 will differ in what should be the transactivating cRNA sequences. So all these are available. All that you have to do is that get the gRNA design and then clone the gRNA inside these vector and the Cas9 sequences. And of course, lot of variations can be thought of in that. And once you do it, you can go through biologistic delivery, and there are some kits are available where you can even have an in vitro assembly. You can have the RNA, you can have the Cas9 protein, and then incubate it with the cells, and then later select for it. But the routine method is agrobacterium mediated transformation, so that is a difficult thing. Like you have to again go through the agrobacterium route. But only thing is, after doing the transformation, like you have carline, you have the seedlings, in every step you have to assess whether the editing is taking place and all by restriction enzyme digestion or PCR amplification, and then do a very, very large, very meticulous backcrossing or selfing to get the agrobacterium vector, TA plasmid vector backbone out. So what is required is only those regions in the susceptibility factor to be knocked out. None of the background should come. So you have to go on selecting and selfing, backcrossing, and see to it that it gets out. And that selection process is very, very tedious, and people have been successful. It's not that difficult. Next slide, please. So this is a whole methodology. So when you come to the work, say for example, if you are targeting the plant gene, I have put here. If you are targeting the pathogen gene, I have put there. Lot of metabolic factors only are contributing to the susceptibility. Some of them are transcription factors, and some of them are very, very simple like halosynthase, and some of them are many of these oxygenases. These are all the factors. Of course, lot of experiments have been done previously to prove that this is the factor that if knocked out, we will get the resistance. and that is background experiments. But once you hit on that, they will definitely develop resistance, and these are the various metabolic genes which have been attempted. On the other side is the genome itself of the virus can be edited, and I have not come across any preference where the fungal pathogen or the bacterial pathogen genome have been edited, whereas a viral genome maybe it is slightly easier to handle, it has been done. So this is the basic principles. Next slide please. For example, if you are going for viral resistance, the elongation factor is the one, you can click it, that picture will come. Elongation factor is the basic factor which interacts with this VPG protein of the pathovirus Y. And that interaction is very, very essential either to initiate the replication or the anchoring of the VPG into the vesicles or the VPG mediated replication. So it is very much essential. But by chance or by scrutinizing the elongation factors, it has found out that there are a lot of mutants and variants available, and in those cases the resistance is found whereas the susceptibility is not found. So based on that this concept came, and there are many institutes in India like IRI, IHR, they are trying to develop papaya ring spot virus resistance using this ELF4 susceptibility factor. Next slide please. So this is the protein. Next slide. Okay. I have more than 4 or 5 slides of tables, but I thought my time of presentation is only 10 minutes. If anybody is interested, I can share those tables because it's in my WhatsApp. I can send it to you. Like usual examples of fungal diseases, lot of it. The tomato disease I will now cover in detail. And in fact in rice, the sweet transporter gene, that has been the major target against for many fungal diseases, and it has been achieved in the case of rice, barley, wheat. There are many such things, examples are coming. And so it is almost like a routine tool now that we can look upon. So this is for the susceptibility factor, that is the elongation factor edited in many of the cases. Next slide please. And you can also, insect sequences can be edited. For example, in whitefly, the white-eye syndrome has been targeted, and it has also been done in many of the thrips and then in arthropods and all, the sex-related or reproducing organs-related genes have been targeted. Next slide. So, the virus sequences targeting, I will talk about this Solanum lycopodium. The slides are coming up, so we can skip it now. Next slide. Next slide. Okay, these are the references that the advantage with the bigomoviruses is that they are DNA viruses only, but the RNA transcripts are being made. So you can either target the replication protein or the core protein or the intergenic region and then make the gRNA targeting them. And such altered or mutated viruses can be used as a pre-immunization in a vegetatively propagated plants, or you have to have that transgenic technology where you are introducing into the plant an edited version of the virus, which itself is a dangerous one. Like at some point or other it may revert. But these are the technologies that are available. Next slide. Yes, this is the example I was telling you for chili leaf curl virus. The work has been done in IRA. You can target either this rep region, CP region and IR region, and these are the gRNA sequences. You can, in vitro, you can assemble the genomic DNA and this assembly and see whether the genomic DNA has been cut or not, and then use it for immunizing the plant. Next slide. So the various PCR for checking up that. You can also do the agroinfiltration assay. Just go on entering it because they have inoculated the gRNA and also the chip leaf curl virus, and they have shown that the virus accumulation is less, the gRNA expression is good, and Cas9 expression is also good. So this is one way to prove all the assay before you really take it up for transgenic strategy. Next slide. Yes, this is a summary of this talk. So this much information, some of them I might have missed, perhaps Dr. Natarajan showed more than mine, it's possible. But I took up these papers individually because various strategies are differing, and some of them are really getting to the diseases that are problematic. Next slide please. Okay, so first is the downy mildew resistance gene. Actually wherever it is a variant, it is recessive, there is an increase in the level of salicylic acid, which shows that perhaps manipulating this gene could help. So this DMR gene was targeted, and this DMR gene again was targeted, and they made the many plants and then they compared by inoculation, and they found out that not only the mildew pathogen but also it gives protection against Pseudomonas syringae, capsicide, which is a spot streak disease, and Xanthomonas species. Next slide. Oh, I didn't like it because the photographs were not very impressive. This is again the powdery mildew, PMR4. They have got 4 single guided RNA targeting this PMR4 gene and made those plants, and these are the results. As you see, compared to the wild, it's not that impressive, but the paper claims that they do have the hypersensitive reaction or cell death reaction whenever you are microscopically examining it. So they say that maybe it has got a potential. Next slide, please. This one is not at all transgenic. It is only incubating the cells and then taking out and then doing lot of screening. So here, in fact, the MLO genes is a very, very well-known genes for conferring the susceptibility across the kingdom. And in this case, in tomato, there are 1 to 6 alleles variations are found. In wheat, 1 to 6 alleles also they did the targeting and they found that it shows resistance. Here they targeted MLO locus 1, and these are the modifications they did in these 2 places, resulting in the deletion. And such a kind of gRNA was used, it's non-transgenic, means either by bionistic delivery or by incubation they could have done it, and it showed resistance to the powdery mildew. Next slide please. Okay, since from morning we have been talking about resistance to viruses, and this TYLCV, the gene TY5, actually it is an RNA surveillance factor. It's a recessive gene, and they found out that one particular line, that 172 line, there is a simple variation in one amino acid sequence, and because of that plant showed resistance to TYLCV. So this is the pelota gene. So what they did was, to that particular sequence, they designed the gRNA and they made that. And along with that, they also targeted the MLO1 locus. So 2 diseases locus they targeted and had 2 types of gRNA, and then again the same processor, and they found out that the TYLCV resistance was very much achieved by targeting the TY5 gene. Next please. Okay, in any such experiments, even in genome editing, there are a lot of off-target targets are there, and if you are sort of knocking out one particular gene, another gene, all those kind of crosstalk between the 2 different type of resistance mechanism, either the jasminic acid based or salicylic acid based, and all those problems are there. So what here they did is, JANS is one of the co-receptors, and it has to interact with the chromatin so that the stomatal opening takes place for Pseudomonas syringae. So they found out one particular allele of jasminic where there was a variation. So they did the genome RNA, the gRNA targeting that particular JAS gene, and they found out that it was showing resistance to the Pseudomonas syringae, and the DC3000 is a very famous strain everybody uses for experiments. So the transpiration rate and the resistance was good, and as an offshoot effect was that it was also showing resistance to the necrotrophic fungal pathogen Botrytis cinerea. Because usually when you develop the resistance to biotrophic, the necrotrophic susceptibility will increase. To balance between the 2 is very difficult, and here they achieved that balance. Next slide please. So the off-target effect can be further met with if you can have a promoter which can be induced. Say for example, if I am targeting virus resistance, the promoter will become active only when the virus infection takes place. Like that you can design your construct. So that is what they made here. They did the Cas9 is come under RGS CAM promoter, which is always induced in the presence of the virus infection. So they did like that, and they made the transgenics, and they found out that targeting both the red gene and the intergenic region, and they found out that there is resistance. Of course, in the CP gene, when they did it, there were some abnormal effects. These are the abnormal effects which are being much talked about, whenever there is off-target effect. Next slide. This is again the translation initiation factor, and they have shown resistance both against CMV and PV1. Next slide. That is again a photographic, next slide. Photographic thing for about PVY and CMV. So they have targeted the intergenic region and they have got the resistance. Next slide. So this is the one that I was talking to you about. Though we know that all the wild germplasms are having the resistant genes, the problem is the genetic drag, and the problem is that to make them, agronomically suitable variety is very, very difficult. We had one L1777, and that was the only one which was showing resistance to TOXADV. But it did not flower at all, and even if the flowers are there, it was not setting fruit. It was very difficult, and we don't know how to go about it. We know that some source is there, we don't know which is the locus, but we could not make use of it at all. But here what they did was, this is a wonderful paper I felt, like they targeted these genes, ovate, fasciated, fruit weight, the multiple flora, and the lycopene beta cyclase. These genes were targeted in a single construct using gRNA targeting all these 6 genes, and then used that particular line because this is Pimpinellifolium, and this has got the source for spider mite resistance and also resistance to many arthropodic insects. So they targeted that. Next slide please. So this is the plant, they got it. I hope you are able to see the tomatoes as good as Dr. Tiku's tomatoes we have tasted. And you will see that the petals number increasing from the single fluorescent, you get the multiple fluorescent, and of course the number of inflorescence increasing, and that is how they land up. So it is possible to edit the wild line and then use that as a source, as a genetic source for further incorporating resistance, and that would be another novel approach. Next slide please. So I concentrated only on tomato papers. There are lots of papers which have not I have not covered it. If anybody is interested, we can discuss about it later. So what I say is, please let's not more and more talk about it. Let's do it. That's what I say. And for all your patient listening, thank you very much.
And basically, the idea is that we thought Dr. Behera would be here to give overview, and she did really well, and I'm very happy about it. Thank you so much. And we will be having more queries to you, and we will take up that question and answer session later. May I request now Dr. S.R. Bhatt, and he will be probably discussing more about potential of gene editing, and I request him to also discuss about ethical and regulatory concern, if you can, sir. That's also an issue which we felt, and a few more questions as and when we start talking about. Thank you.
First of all, let me thank the organizers for giving me this opportunity. I'm no expert in genome editing. Only thing is my association with RCGM, and in the formulation of guidelines, I had a role. So, in that capacity, maybe.
Disturbance. Without that also we can hear. Put it up.
Put it up.
I think I'm audible.
Yes, very well audible.
Yeah. The topic that I've been given is to discuss about ethical issues, regulatory challenges, unintended consequences, ecological impacts, and a small question whether we can distinguish the gene edits from the conventional mutations. Before I discuss this, let me just say about the technology itself. Genome editing came at a right time, I would say, particularly the gas-based, because we had lots of information about molecular biology. We also had accumulated enormous information about genomes, sequences, functions, and lots of things. People were just wanting to do it and see whether we can manipulate it into it. And that is the reason, the moment it came in 2012, it was immediately picked up by both plant science people and animal sciences, particularly the health sector. The chief advantages were the precision. You could do exactly where you wanted to do, what you wanted to do. Efficiency, very high frequency of editing at the choice. The most important is time. What would take years to do, even in the model systems, we could do in months and weeks. That's how this technology picked up enormously. And it also offered unique opportunities to tackle some of the problems, particularly in the medicine, which are intractable practically. Many inherited diseases, for example, thalassemia, beta thalassemia, sickle cell anemia, muscular dystrophy, for which we knew sufficient genetic information. We knew the gene, we knew what exactly the amino acid chain, but we had no means to correct it. This gave an opportunity. In fact, sickle cell anemia, the cure is likely to be coming very soon, because some of the experiments, people have shown that they are free from any requirement for a couple of months, even years. So its impact in the medicine is far greater than what it is likely to be in plants. Fortunately, in plants we could do anything and everything. In fact, there is very little ethical issues when it comes to plants. We can do whatever we like. Practically, we have been doing that in the conventional mutation field. So, these days what is happening is technology is outpacing even comprehension. We do not know what are its implications. Forget about how do we regulate about it. The latest would be this chat GPT and these AIs. We do not know. In fact, in the science field, now chat GPT can write your manuscript much better than what we do. And every journal is facing this problem, whether it is chat GPT writing, submitting a manuscript, or is there any real author behind it?
Including thesis, sir.
Including thesis. Even Nature and everybody is saying. And many of the authors are now admitting that they have used it, because subsequently they have prepared a rough draft from that and they have made a minor genome editing there. so people are coming forward with that. So considering this, see, CRISPR as opposed to the traditional, let us say, nuclear science or space technology, which would require lots of teamwork, enormous infrastructure, and years of effort. If you want to send something on Chandrayaan, it would take years of effort. Whereas CRISPR is something that could be done in your backyard. In fact, today, CRISPR tools are available. You can order in the US, and it is being used by the children for school experiments. So, every technology has dual use. Something could be used for good, and invariably there will be many dangerous uses of this technology. So we have already demonstrated that the Chinese, he has produced 2 children, twins, by genome editing, in spite of all the bans that we have. And he has announced it in a conference, and he is behind bars right now. But still, can you imagine somebody doing that sort of a thing? But it happens. Scientists do it. So that's the danger of this technology. In fact, Jennifer Doudna, soon after they made this, she organized several Napa Valley meetings. In fact, Paul Berg, whom I had a fortune to meet him personally in Stanford, he was the one who did the Asilomar conference, if you remember, in the 70s. So she contacted him and they devised this, and they put up certain guidelines as to what should be done, what should not be done. Well, fortunately, in plants, we have greater freedom. There are no big issues in what you can do, what you cannot do. Whereas there are strict guidelines in the case of humans particularly, whether germline editing is permitted or you can do only the editing of individuals so that they do not transmit further. And whether cosmetic changes should be allowed in humans, that is prohibited. In fact, even in animals. Animal biotechnology is not, genome editing is not free from all the regulations. They are still considered transgenics, even in the US, where everything else is liberal. But for animals, they are still kept it as separate. So we are lucky in that respect that plant people are allowed to play, and let us make use of this opportunity. In fact, when we were asked to develop these guidelines, Dr. Prabhu was the chairman and I was one of the persons. We said that, see, we will prepare the guidelines, but you make sure that it will come through. Because many times we prepare guidelines and nothing happens. But fortunately, Government of India was very keen and they accepted entirely as we had given. And we have kept the minimum sort of regulations as far as possible. The local Institute Biosafety Committee has the overriding freedom to let the things go. Unlike the earlier versions where we had RCGM, GAC, things would come back and forth. This has been made very simple, particularly with regard to the SDN1 and SDN2. Practically, one can do most of the work at your laboratories and get out the products as far as possible, as quickly as possible. And we feel that the SDN and SD2 are the ones that most of the people will be playing with. So, with these backgrounds, let me just take up some of the questions that you had mentioned. Whether we can distinguish gene-edited products from the conventional mutations. Yes and no, both. From our experience, the conventional mutations would lead to lots of variations at different places. Okay, for example, in one of the recent genome killing by sequencing work in Brassica, we have found that on average, an EMS mutant contains more than a dozen mutations in different genes. So if you do genome editing, you would not find so many. But this is only theoretical. If somebody does not say, and if he just crosses with a mutant and says that this is a mutant, there is no way to find out. So this is the ethics that we have to follow. If you are honest, you would admit that you have done genome editing, but if you are dishonest, we cannot catch you. Certainly we cannot catch you. And some people might use it because you would be free from any regulation. And also you might be free from any patent also, if you say it's a mutant. So we do not know how people will take it. It's not possible to regulate that part of the story. Some of the concerns that I have personally is how equitable this technology is going to be. Tomato, fine. Genomic resources available, you know the genes you want to edit, but what about other crops? We might see a scenario just like Green Revolution, which was limited to a few crops. A set of farmers benefited and the rest of them did not get anything. So which would create a disparity. And also some crops which are highly advanced in terms of genomics would do so well and displace other crops. This is what will happen to the millets. We say that, oh, they were neglected. It is the success of corn, for example, which has wiped out so many crops and is continuing to. Because you can use it for anything, because when there was overproduction, you said you feed it to cattle. When you still had more bioethanol, corn syrup, this is how corn overtook everything. Similar thing might happen if genome editing is, I won't say exploited, is used by those who are in a better position. How do we really see that the crops, like one of the areas which is most difficult in terms of breeding is the perennial fruit trees and crops. Genome editing has a great future to improve those things. But it's the least understood of all the things, including the transformation. Genomics, nothing is there. If you want to do, let us say coconut, mango, any of these things, we have nothing to do there. But the most benefits could come from there. This is one problem that I see which has happened in the past and likely to happen with this technology also. Some crops will remain orphan, will keep neglected, although they would require the most attention. It is like some of those diseases which occur only in third world countries or occur in very small frequency. No pharma industry wants to put money there. In fact, some of the people are coming forward now to do it themselves for some of the human diseases because their children have it. They say that we have very many good examples of that sort of people who have taken it upon themselves to do research and develop solutions. The other concern that I find is that a handful of genes will be edited by everybody in every crop. The best example is herbicide tolerance. Everybody will put herbicide tolerance in every crop. Then what will happen? One thing is either herbicide resistance will develop in the weeds. Secondly, overuse of chemicals in agriculture. Because every single crop. Can we avoid this? Is there a solution? Or we might run out of the herbicides also. So this is another concern that I see. Off-target editing, lot of people talk, but the experience is off-target effects are not going to be serious, particularly in plant. And even if they are there, we can segregate them out. It's not a big issue. Unintended effects. It's very difficult to say unintended. What is unintended? In a sense, it's only when it manifests that, oh, I didn't want to do it. But there is no way to guess what is the unintended. And it's very difficult to design a set of experiments because we always used to have this problem. I was in RCGM for a long time. You cannot really do any experiment for unintended. Because you do not know what is that. So this is something that I do not know how to make it. But what is happening is, for example, making a plant to grow in inhospitable conditions. Let us say salt or this and that. What you will make? You will take the crop to places where it would not grow. And it will create a new scenario of pests. And also, we will be able to make crops grow round the year. Like remove photosensitivity, cold sensitivity, this, that, all this could be done. Then what happens? The crop will be grown round the year. Your pest dynamics will change. For example, I will tell you an example recently we came across. Soybean. Soybean is being used as an intercrop in sugarcane in Maharashtra and Karnataka recently. It gives a very good yield, soybean, without any loss to the sugarcane. But that makes it almost round the year crop. And that is creating a problem of pests. These are so-called unintended because we never wanted to do that, but it happens. So these are the sort of scenarios that are likely to come because we have the power to modify most of the things. Things can grow in places where they should not grow. And it is like, I have a technology, we are all comfortable. Let the temperature be 45 outside. We can have ACs and this. But ultimately, the heat is around and it will affect all of us. So this is one of the concerns. Also, GABA is given as the first example of nutrition. Okay, we can make nutritionally rich crops of various kinds. It could also be equally nutritious to the pests. and Massachusetts. So, are we going to tackle that, and how are we going to? We have not seen that part of the problem. That will it make it more susceptible by having these sort of nutritional factors? One of the examples that I can take from conventional mustard is glucosinolate free meal, and genome editing is very successful. Delhi University has done it, and multiple genes. Conventional breeding is very tiresome, but it could be done at one go by this one. But do you know that glucosinolate free seeds are highly susceptible to insects, pests? Every bird will watch and find that particular this one. Even if you sow, the peacock will pick up only that line in the entire field after sowing. So similarly rats, and it's keeping quality. So while we can make it, these are some of the unintended effects that can come in the course of this. And lastly, Dr. Malti was very attracted with the neo-domestication. The consequence of neo-domestication would be that if you have made Pimpinellifolium, there is another wild species which has been
Chinese.
when neo-domestication was
There are
done.
several.
Yeah, 3 papers came simultaneously. Now, gene flow with the wild will happen very, very easily. What will happen to gene flow in that? What will happen to the wild species diversity? There will be non-shattering, for example, at one of the key domestication traits. So, the seeds may not disperse properly, and also the fruits being larger and other things, this number of seeds in the wild would be less. So these are the sort of questions that will arise, and we have to really judge these consequences very carefully. In fact, we are not talking about gene drives here, which has a great potential again in the case of insect management, but it's equally risky. Gene drives could be very successful, but it could wipe out the entire biodiversity. These are some of the thoughts that I thought I should share with you. Thank you very much.
Hello, sir. Good afternoon. Yeah, thank you.
We can't hear. Volume. Connected to the. Volume.
Can you check the system volume?
As the previous speakers had said, I was really half qualified to talk on this subject because I have almost experience with the legislation. Yes, we have taken beauty cotton and beauty corn through the regulatory pipeline in India for the last 15 years. However, if you look at the global scenario, all the countries who had legislated the guidelines for genetically modified crops, GM crops, quickly got up to the act and they deconstructed the entire GMO regulatory guidelines, and they found the task to be much easier if they had adapted genetically edited crops. And so if you look at all the draft the legislations of all these countries, there are a few consensus points which come out very strikingly. One is, all of them agree that genetic changes introduced by SPN1 and SPN2 types of CRISPR-Cas9 technology do not differ from the changes that occur naturally or those from conventional breeding. This is a kind of common thread which runs through the drafts of all these legislations. And you see, with genome editing in the last 1 decade has been increasingly viewed as a modern breeding tool that will modify the gene without leaving a piece of its DNA fragment. So this is a very important assumption all of these draft legislations have taken. And the legislations very, very specify that no extraneous DNA should be left behind in the plant or which has been genome edited. Now, in this exercise, some of those countries which have been very tough on the guidelines, they have also created avenues for including gene edited crops, exempting it from the GMO guidelines. For example, they have tried to finely differentiate between transgenesis and cisgenesis. And thereby, they have opened an avenue for the transfer of genes in the same sexual compatibility group. Probably in future, this changed thinking would also bring in STM3 mode of genome editing into the exemption clause from GMO guidelines. I mean, this is only hope. Now, most of the countries in their draft legislations, they have redefined genetically edited crops, and even they have attempted to reclassify genome edited crops, and they try to bring them under varietal registration or seed regulation, much easier. And they have also tried to redraft the food safety guidelines to include and clearly differentiate between genetically modified food and genetically edited food. Now, environmental risk, food risk assessment guidelines have to be framed in view of the differences between GMO and genome edited crops. Even if you look at the guidelines, given out by Government of India, they too have exempted STM1 and STM2 from the regulatory guidelines of GMO. However, they have said that they would have to go through certain safety risk assessment studies. Now, let's look at the concerns. While I voice the concerns, some of the areas might overlap with what the previous speaker had said. One concern voiced by almost all those countries has been off-target edits in the plant genome. But we all know that the current advances in gene editing show convincingly that the probability of off-target changes or mutations are no more than the rate of spontaneous natural mutations which occur in nature, which is in the range of 1 in 10⁻⁶ to 10⁹ base. That is the probability of a mutation occurring naturally and spontaneously. However, they also admit that the off-target mutations through gene editing would be much lower than what we have seen with chemical mutagenesis or radiation mutagenesis. So, yes, I think the path for gene editing regulation will be much easier in future. Another concern is the detectability of the modifications which have been made in the gene edited crops. Like my previous speaker had said, can you detect a gene edited crop, especially by STM1 and STM2? He said the answer is yes and no. Okay, so I'm going along with the same answer. Now, however, if you look at the commercial regulations, this could impact the traceability of gene-edited crops and their products. Now, this could be very important for those countries which import gene-edited crops but will not grow them, because they have to come out with a detection mechanism. And detection methods will have to be made which will distinguish natural mutants from the gene-edited ones. I think this is a far cry. I mean, it's very difficult. But again, these are some of the requirements which probably we might have to meet. Now, and also the ability not to distinguish between natural mutations and gene-edited mutations will also pose several questions, several problems in courts of law. Now, what are the alternatives? For example, if I am to release a gene-edited crop, well, as per the Indian regulations, it is enough if I go through the IPs, the local IPs. But do I have to make a label claim? Do I have to self-declare that these are the mutations I have brought about in the crop? Yes, I think that is what is expected. But then the question is, how far can we rely on the label claim or self-declaration if in case of crops which have multiple edits? And what if there is an admixture of gene-edited crop of one type with gene-edited crop of another type? I mean, these are some of those very difficult questions we might face. And will food products resulting from gene-edited crops, will they be labeled on the shelf? This is a very important question because, scientifically we might say that gene-edited crops are no different from natural mutants or, brought about by breeding techniques. However, if we say that and do not label the gene-edited product, that would be infringing on the consumer freedom. And I think that is not right. So I think it's part of a very difficult situation as far as the commercialization of gene-edited food is concerned. Now another issue is, as a direct follow, will there be disparity among farmers? Because after all these gene-edited crops which come out from about 3 companies, the license, they will be a trade fee to be paid by the farmer by the seed. Now, if some farmers cannot afford to buy the seeds, there is a disparity between those farmers who want to adopt the next-gen technology and those farmers who cannot afford to go into the next-gen technology. We will be creating a disparity unless gene-edited crop comes out from public-funded institutions like the CGIR. Now, the legislations, the draft legislation brought out from all these countries should also, contain a clause which will support small and medium entrepreneurs, in the developing countries, to enter the market of gene-edited crops and contribute to maintaining or even expanding the diversity in the seed market. Because seed market is also equally important as the food grain market. So as I said, public-funded institutions like CGIR Consortium and others are the only hope for the democratization of gene-edited crops in the future for the farmers. Now, as far as patenting is concerned, I think the foundation patent of CRISPR system was awarded in the US to the Broad Institutes, comprising of Harvard and MIT in 2014, after which many medium and small players have also applied for patent, and the Indian patent scenario constitutes about 3% of the global patents applications as far as 2021 is concerned, and many of these patents are in the healthcare sector. And so, still, of course, some institutions have got the right to license the CRISPR technology to users, but still the scenario is not very clear and it is very hazy. And if you look at the countries which have stepped up to embrace gene-edited crops, the naysayers to technology are only 3. That is New Zealand, South Africa, and the European Union. Though European Union is not very hard on the gene-edited regulations, I mean, still, as in today, it has to go through the regulatory pipeline of GM crops. However, they are willing to review the scenario of gene-edited crops and the new tools which come into the market every year, and possibly they might open up their markets in the near future. So, summarizing, the countries which have embraced and which have got the legislations in place are USA, India, China, Australia, Japan, Brazil, Sweden, France, UK and Chile. They have exempted gene-edited products from the GMO regulations if the final product does not contain a transgene to give agriculture.
Yeah.
Thank you, I'm done.
Thank you, Dr. Mohan.
Everyone, and thanks for the opportunity. Thanks to the organizers for organizing such focused workshop on genome editing and genetic engineering for tomato improvement. So most of my previous speakers, they have touched upon all the technical aspects. Just I want to sensitize the audience on few of the points that they have not touched upon. So there are so many positive positive things or advantages of using genome editing in accelerating the genetic gains. So when we look into the history of plant breeding, so we are not new to mutation breeding. So it's there since 1900. So when De Vries coined the term mutation. So but when you look into the problems associated with those methodologies, so either any of the artificial mutagenesis methods creating novel variants, chemical or physical or molecular methods using pDNA insertions and all. So you will get the variant, but the problem was like unintended targets. So in our own example, I have seen like even by using EMS mutagenesis, we came across more than 50,000 mutations between the wild type and the mutant. So base pair substitution alone, when we took the index, so it went around 5,000-6,000 index. So you will get undesirable effects or unwanted mutations in the genome. You will not be able to control the mutations or mutation rate. In addition, so the desired mutation, obtaining or identifying or targeting, achieving desired mutation, so it's a number game and also our luck. So if at all we want to identify a rare mutation or generate a rare mutation, you may require a lot of resources. So just for an example, so in our own experiment, so when we invented or developed the India's first herbicide tolerant mutant using EMS mutagenesis, we identified one plant in 300,000 plants, F2 progenies. So such a rare event it was. So we may sometimes, if we don't go up to that level for screening or developing the population, we may miss it or we may not be able to achieve the desired target. So this is one of the major lacuna, and that particular major disadvantage we can overcome by using this CRISPR-Cas mutagenesis or genome editing. So there are n number of versions. Now we have very user-friendly CRISPR-Cas9 systems that we can target any gene, we can generate or we can recreate any mutation that we want. In addition, we will be able to create any novel mutations in any gene. So that is the power of this technology. So novel ideas, we can, in our own example, in one go, so in a span of around 3 to 4 months, we were able to generate 23 different alleles of the rice aromatic gene called DADH. So such a powerful technology is there. But there are few concerns, so particularly in tomato, so on using this technology for improving its efficiency. One is already highlighted by the previous speaker, so Dr. Bhatt and also Dr. Kopan, that off-targets unintended mutations. But now Cas9, it is known for its high frequency of off-targets. But now recent versions of Cas9, Cas version like Cas12a, and then other improved methodologies like prime editing or base editing, and then the independent Cas9 without PAM sequences. They are all there, so to specifically target the gene. And then few other concerns like cytotoxicity of Cas9 needs investigation. And in terms of delivery, like we need other tissue culture independent methodologies we need to develop. And then in terms of base editing and prime editing, so we have only the transition mutation possibility. So we need to think of developing the system which can help us to generate transversion mutations. So these are all the few limitations or challenges with these technologies, particularly tomato is an 80-rich genome, so which will have challenges in achieving desired changes through mutation. So we need to generate large number of mutant progenies or even to achieve the desired mutations. And the regulation wise, particularly Dr. Karapa has asked me to talk or indicate few things on collaboration.
Yes.
So collaboration, so it's very much essential in any scientific innovation, so or scientific research. So working in isolation or will not allow us to reach the target in a desired period of time. So in our own experience, we have learned so many things through collaboration. It will help us to learn new things, new knowledge from our collaborators, and then it will help us to avoid mistakes so that we will be able to reach the targets in a lesser period of time, and we will be able to learn from others' mistakes, and in turn this will help us to improve our own efficiency. And then the collaboration should be an open channel. There should not be any restriction so among the partners to exchange the material, sharing the data. So we need to formulate the program in such a way that all the partners are able to communicate and exchange freely. And then finally, in my experience, I have seen in several cases that network project or collaborative project, so we were able to move faster and we were able to accelerate the outcomes. So in a shorter period of time, and based on that, we have suggested so many things that is becoming very common today. And the crux of the success of any collaborative program relies mainly on defining the roles and responsibilities of the partners in the issues so that it will be a open platform. So these are all few of my opinions on this topic.
Thank you, Dr. Ravindran. We are a little bit hard pressed for time. We will come back to you with specific questions if any.
Respected delegates, very good afternoon. Myself, Suresh Reddy, from tomato breeder from IIV Water. Why our beloved director was interested to come here, he booked tickets because of feedback. He was not able to.
Switch it up. Switch it up. Yes. We can hear you without mic here.
Yeah. Also, we can ask that is.
That's why.
Yeah.
Okay, go ahead. Sorry.
With respect to this technology, I had an.
Come here. Speak little louder. Then we can all hear. Come over here. Center badge. Yeah. Yes. Now we can hear you.
With respect to this technology, I had an opportunity to get training in Australian National University under DPT sponsored fellowship. Before going to this aspect, I want to add one addition which Malti Madam was telling to when we go for genome editing. Before going for genome editing, when we guide RNAs, we should know its efficiency, whether it is able to do our desired double stranded cuts in our targeted genome or not. Madam suggested to go for in vitro method. In vitro, in addition, because I have worked in that, I want to add, there is one agrobacterium rhizogenesis species like tumefaciens. Rhizogenesis is also there. Within 3 weeks, in explants we can get more than 1 centimeter of that root system, which we can isolate DNA, and we can check whether double stranded cut which our intended genome editing is happening right now. That is one of the addition I want to do. Then with respect to education and public awareness, already we know this technology has a potential to solve many human challenges, especially in agriculture also. So this technology is going to stay. We are going to work with this one. But whenever new technology comes, we have seen, especially with case of COVID vaccines, there will be so many rumors around it, which hinders acceptance of technology in the society. So there comes public awareness, to bring public awareness and education of the society and the different groups in the society. For that, what we have to do, as we do for other technologies also, we have to educate people, especially with respect to what is SDN1, SDN2, 3. This we have to do as this technology is going to stay. First, we have to bring them in curriculums, education system. Then, in all levels, like from secondary school level to preschool, pre-university, university level. Then bring out so many competitive like projects, discussions and discussions like panel discussion in public, especially with we have to bring farmers, KVK subject matter specialists, and more especially pressure groups, which we have seen with the case of genetically modified crops. We have to convince and we have to bring pressure groups into the scenario wherever we are going to bring these kind of genome editing discussions. In addition to that, with respect to other things like regional languages, because Indian society it is has variation with respect to many things, especially language. We have to take it into regional languages. So whether it is newspapers like in Telugu, Annadata is there, and like that in Maratha, Kannada, different regional languages are there. We have to take that one, whether it is radio talks, TV talks, those things. In addition to that, involvement of organizations like Biotechnology Consortium India Limited and Center for Public Understanding of Science. These are like non-governmental, but they are they have, whenever Nobel Prize comes, they are bringing some kind of in simple language which even normal men can understand. They are bringing what they have contributed. We have to bring them, we have to rope up them into this system. And the utilization of social media, we cannot ignore social media in this. So YouTube channels, wherever possible things, we have to educate through social media. Then second question was like, how can government and public sector institutions facilitate the adoption of gene technology by small and medium entrepreneurs? Yeah. With respect to these technologies, when we use, until we prove, even in SDN1 and SDN2, we have to prove that foreign genome region is not there in our product. Until that, it is to be under containment conditions, even though it is under institutional biosafety committee as sir told. Until that, it should be there. For that, we need technology, facilities, which small and medium entrepreneurs cannot offer. So how we can facilitate them? For that, already institutions will work on technologies and they give genome editing products. So that as normal varieties, lines we are taking, private companies can take those licenses and use for commercialization. If small and entrepreneurs, they have new idea, they can come with funding or on PPV, they can float projects in public-private funded manner. And there are public institutions that are providing hand-on training, workshops on different aspects. Here I want to mention National Genome Editing and Training Center, which is established in Navi, Mohali. There they have exclusively one center for training the people on genome editing. This shows that organizations, institutions are how accepting this technology as future technology. Then one more suggestion is like private, whether different labs, they work on suppose one genome editing aspect. So in that clones will be there. Suppose we want to develop pelota resistant by editing pelota. Suppose our lab, we establish that clone, we clone that one and we put in Cas9 background and we develop binary vectors, and as I told, rhizogenesis is there, agrobacterium, tumifaciens is there. And these all clones, once we do product, there should be some repository like add gene. So we can deposit there. So whoever interest, for them no need to design again guide RNA and do all kind of exercise. Directly they can contact them. There will be definitely some kind of formulations, regulations which should not be exploited in wrong way. That should be there.
Thank you.
Yes.
Does it include field trials also?
Yes. See, IBSC conducts individual this one.
Yeah.
Till the time that you have shown that it is free from backbone and any extraneous, till that time it will be IBSC. As you do for the transgenics, till that time it will be handled in the IBSC as a transgenic.
No, for field trials.
Field trials you will come only when you have the backbone removed.
Yes, see, I'm ready with all that backbone removed.
See,
but for conducting field trials, you have to go to RCGM.
Field trials in the case of conventional transgenic also, it's the event selection trial is the earliest field trial that one would go for. Here, event selection is not a question. You can do this in your confined, contained net house, glass house, whatever. So once you remove the backbone and establish that it is free from that, you submit that application, then there is no question of regulation further.
Okay. It's not regulated.
It's not regulated.
This information. What is sorry, I should go in between. It is just information to RCGM. I mean,
It's information.
But so far we have not conducted any field trial of gene edited.
No, that's
so
not
far in India.
So it will come. It will come very soon.
But still we do not have the experience.
No. IRI has some events. Yes.
Not in tomato but in rice.
Rice. Dr. Vishwanathan. Yes.
I have one question to the policymaker, maybe an advisor to the government. This technology is getting popular, and then we know that it has a lot of potential. But then where is the technology licenses? It's a very critical issue. And is government taking any efforts to get common licenses and everybody can access it? I mean, that's what I want to know.
Yeah, Dr.
This is a grey area. So far, there is no consensus. The government of India had initiated some efforts to negotiate, but they say that because it is still not sure who is having the ultimate patent.
Yeah.
Patent is still disputed, even for the medical. This one, Corteva has this in India.
Corteva has
this one. They say that right now it is available for free for research, but commercialization they are also keeping silent.
Yeah, but then companies
And also, it is not only the patent with regard to the technology of gene editing. Every gene that you are thinking to modify has its own individual patents. Whether patent is there in India is something that we will have to check. Because it's possible that India they do not have a patent, then you are free to use it. That's it. Every single gene that we are planning to, whether it is elongation factor or it is
outside India, it's there.
the people have already patented it, whoever has discovered it. So most probably they have not put a patent in India, so we might be free to use it.
Yeah, but then this is the grey area and very
This
serious.
is a grey area. Only when you reach there you will know. And they are also very clever. They know how much money this is going to make. Accordingly, that's why they are not agreeing.
Label it is as a gene edited crop, they will.
So that's why I said there is no way if you say that it is mutation. Only thing you have to have some experiment to show that you are doing mutation.
In fact, that was my question, Dr. Bhatt. Even to say that none of the backbones are there and all, who will be performing all this parameter test?
No, you have been asked to, in most cases. No. There are two ways that people are asked. One is that you know the vector. Every single piece of the vector will be PCR amplified and sequenced. That's one of the easiest approach. And now that you can also do whole genome sequencing, you can also do both.
Better.
So on a small scale, PCR amplification of the entire vector. When you have the final, this one, then you can opt for, depending on the genome size. If it is wheat, you may not.
No, but some people deliberately want the Cas9 to be incorporated and to be editing the gene permanent.
Oh, that's a different purpose.
Purpose.
That's a different
Ma'am,
purpose.
regarding that you said we can develop virus registers using virus targeting
Yeah,
one.
yeah.
But that won't fall under this category. That is transgenic. You have to keep on expressing Cas9 and HGRN.
That is transgenic.
No, it's not HGN3. It's transgenic.
It's transgenic.
GMO.
It is GMO.
Because you have to continuously express
You have to keep it there.
HGRNA and Cas9 in the plant. So that makes transgenic.
See, the other area, because there are a lot of breeders here, where this one would be very useful is in the production of maize sterile lines as well as haploid inducer lines. Those mutants, if you can really do it, it will not be directly for the breeding, but it will be useful for your development of varieties. One of the things about what we had in mind when we listed out some of the things to be discussed was the collaboration. See, there are big companies, small companies, resourceful companies, less resourceful companies, and all of them have presence in the market, maybe big or small, but each one of them would require genome editing at some stage or the other. This is where we need collaboration.
Come together.
Collaborations, if you have a good genotype which would require genome editing, somebody should handhold those small people to make it happen. I do not know whether it is possible, because even though we say that it's very easy to do genome editing, when it comes to doing it and taking all the regulatory issues, it is not feasible for each and every company.
Consortium approach will not help?
Consortium will help, because sometimes people would say that I want to keep my variety. I would only request you to do this.
That will be the problem.
That will be where the problem is. Although there are people who can do it. In fact, one of my students has opened his own company. He is able to do genome editing for anybody. He has no business in the seed. But he can do it. He can do it.
As he stays within that framework.
Yes.
But still
He doesn't want to go into
But still he needs license, sir.
He has
In India, sir? In India?
in India.
In India.
In India.
Any specific queries from the audience? Dr. Ramon.
Yeah,
There
sir,
is
a couple of questions.
a-
You already mentioned that there are perhaps a couple of applications with the RCGM where they have received intimation that IBSC at level the vector backbone is clear, they are ready.
Only two instances.
Two examples. What is the status of it? In the sense that it is done deal, that they are ready to.
No, see, they themselves would require, increasing the seed and these things. They have just got the events and made
So
sure.
they need clearance from
So my
license
question
holder.
is, eventually, say 6 months later,
Yes.
is the stage where they perhaps want to take to the land.
They will.
Yeah.
They will.
So, do we anticipate a 2009 BT moratorium scenario? Suddenly activists coming into picture, and are we, as a machinery, are we doing something in anticipation of that, or?
No, absolutely right now there is no such thing, because
The
we
answer is yes,
Because
it's
there are-
going to happen.
take it for granted, as he said. Yes.
Yeah.
Yeah, because there are 2-3 things which are
भाई
sticking.
डिस्कस कर रहे वो यस है
Huh?
वो यस है
डेमोक्रेसी में आएगा. डेमोक्रेसी में आएगा.
We already.
अब मैं I don't want to say anything more.
So you already mentioned.
You take the
that-
message.
Especially
That's
for
all.
Karnataka.
Yes.
Sorry.
Activists
No, you already mentioned that it's a grey area as far as access is concerned. So we are also on the sticky stage in terms of infringing on the patent rights. And we also know that it is very difficult to differentiate this versus the conventional.
Yes.
So eventually there is really no respect for, if you really want to, somebody can just pass on the material.
They can.
So with this kind of complexities, don't we expect the pressure group to be at a higher pedestal than earlier? And I think we are just talking about the potential and how big
True.
is the opportunity. But I think we are missing, if we don't have a system in place, I think we are missing a bus.
See, the question is that we will cross the bridge when we reach there.
Correct.
That's all.
Correct.
There is no way.
Yeah.
Don't have to imagine.
Organizers request us to complete everything. 3:45, now it's 3:45, so we have to conclude. And there will be definitely many more queries and questions Probably you can write to us, right? So we will try to share the queries, or we may request our panelists to help us to address the questions. So that's how we can do it. I would request Prakriti to conclude and share
If
the queries.
ATPBR, I have a suggestion. If ATPBR has any regular sort of query where
The good companies will use and make money. The smaller will
Dr. S.R. Bansal, Dr. P. Fakhruddin,
That's what. My big ones can.
Dr. Nataraj Karwasar, and Dr. Y. Suresh. I request you, sir,
Like for instance, the
Develop ID for that is not a problem. Like for my vocation, we can develop identification essay. For particular plane, I can develop essay. But then how I am going to challenge him that it didn't come from medical education?
Everybody should.